Hi, this is Celeste from the Celestial Report, and today we are going to be talking about Operation Adam. A lot of us have had indications that there were pieces of a puzzle, but we could not put the puzzle together. And I had a piece of information and I did not know the two definitions. So I looked into the two definitions of these words that I didn't know, and I tripped into this whole world of Adam. And it is clearly an operation, which you're going to see in a few minutes. And it's something that is basically, it is the playbook. It is tying together all these loose pieces that we have. And so I'm going to share my screen. And rid of that. And let's, okay, let's start. Sorry, it takes me a minute to get organized here. So this is Operation Adam, the dark remake of humanity. And I am going to be stopping at various places within this presentation because some of the information that I am going to present to you is technical. It's more on the medical side. I've done my best to eliminate the highly technical words, but there still might be some questions. And so it's going to give you an opportunity to ask those questions. I'm not going to answer emails. So this is your opportunity to ask these questions now. So I'm doing this with all humility. I strive to relay to you this really convoluted operation named Adam. It's anything that the enemy does is very complex. And so it's not such an easy task to unravel it all, but I'm doing my best. So these are the words of a gamer who knew biology and with great accuracy described what we are facing. Adam is a canvas of genetic modification, but plasmids are the paint. What is Adam? Adam is a chemical substance that can rewrite, tongue twister there, genetic material, allowing the controller to alter bodies, genetic makeup, and natural abilities without any direct limits whatsoever aside from their imagination. Adam does this by supporting the formation of potent stem cells that can shapeshift and morph into any cell type, including a variety of unnatural ones. The type of cell formation, however, is also compared to a benign form of cancer. The chemical slowly destroys native tissue cells and replaces them with unstable stem versions. And this is what we're seeing in the editing process. When did biological experimentation earnestly commence? So Darwin published his theory of evolution with compelling evidence in his 1859 book on the origin of the species. By the 1870s, the scientific community and most of the educated public had accepted evolution as a fact. Around the same time, Helen Blavatsky began espousing the Blavatsky theosophy, the synthesis of science, religion, and philosophy, proclaiming that it was reviving the ancient wisdom that underlies all the world's religion. That's what I believe is fallen angel theology. The operation began early in the 20th century with biologic experimentation of children. These children became autistic. The development of autism in both frequency and the amount of money is not classified, but it can be found, some of the money can be found embedded in a precision medicine timeline at this particular document. Biological experiments are more numerous than the sands of the seashore. In this presentation, I am just going to focus on autism because otherwise it will get too complicated. So does anyone have any questions at this point?
There's no direct questions relating to this yet.
Okay.
Except one, sorry. Do you know any being that represents Adam? So being in inverted commas, do you know of any being that represents Adam?
Well, we represent lowercase Adam, as in mankind. Is that what you're referring to?
Well, we don't know. That's just what was written here. I haven't got interaction with that person at this point. That's the question. Do you know of any being that represents your
Okay,
Adam?
that's what we're going to be talking about. So you might gain some clarity as we proceed. Okay, so let's do a little bit of a mini deep dive into the Autism Project. Autism is thought to be a developmental disorder characterized by the following severe deficits. And as I read these, I want you to think about where society is, and especially our children, although that it can be anybody. This is pervasive. It's not only autistic children. We have created a culture of autism. So there's limited communication. I remember one time I was sitting in between two of my grandchildren. They both had cell phones. I did not have a cell phone, and they were texting back and forth. And I had not seen my grandchildren in some time. And they never said one word to their grandma. They just carried this conversation on. So it's very limited communication and a very divided communication. Extremely limited range of activities and interests, repetitive and stereotyped behaviors, inability to engage in normal social interactions, and we're finding that more and more today. Intense self-absorption, that's the whole selfie generation. Language dysfunction, and disregard for external realities, such as what is happening with our world. And then the graphic of the symptoms of autism give you some other characteristics of autism. Now, this is what I found was very interesting. The first case of autism came 1 year after the first blood transfusion in 1907. So the first so I think the transfusion was in 1906, and the blood transfusion was in 1907, and the first autism was 1 year later in 1908. And until 1980, autism was considered a subset of schizophrenia. In the days to come, you are going to see many links between the SARS-CoV-2 and schizophrenia, especially as this neurological warfare hits a crescendo in 2022. So we must briefly follow the trail of autism to understand the touchstones of the biological war against us and why current genetic edits are decimating all biological life as we know it. You may have noticed the current generation has some autistic symptoms or manifestations. The definition of manifestation is to make apparent, as in someone manifesting a demon. The tech giants, in coordination with each other, are systematically dumbing down the population by aggressively, biologically mutating both the body and the mind. Please notice that the first autism did not occur until after that first blood transplant. Before 1908, autism was unknown in human history. So I'm going to give you a brief timeline. It may seem irrelevant, but it really does give us an idea. It gives us one snapshot into this huge, complex genetic modification of humanity that's going on. So in 1907 was the first blood transfusion. In 1908 was the first case of autism. In 1911 was the first recombination of genes, which really surprised me. In 1942, epigenetics describing the mechanisms inherent to the process of modifying gene expression, known as phenotypes or clades, which is directed evolution. 1945, DNA as a driver of proteins was discovered. In 1955, the refrigerator mother theory identifies autism as a psychological manifestation caused by unexpressive and unloving parents. Now, this is about the time that the UN was really ramping up, and they used to call their conferences and their little get-togethers manifestations, and it really had quite an evil overtone to it as they were discussing how they were going to implement this biological war and the change of humanity. In 1960, there was the first supercomputer. In 1961, the genetic code was cracked. In 1964, autism results from environmental assaults that triggers a series of biochemical defects. In 1965, the Autism Society of America was founded. And in 1967, the Autism Research Institute, a nonprofit, revenued $1,754,803. In 1970, Temple Grandin, and maybe many of you are familiar with her work, you can find her on YouTube, advocates for autistic people, but links them with animal rights. And that is all under the One Health umbrella. In 1970, the pharmaceutical treatments for autism begin. In 1977, twin studies concluded that autism is highly inheritable, with environmental factors accounting for only about 10% of the cases. So this begins the twin studies, much like the twin studies of Nazi Germany. In 1977, the patient's right to decide was passed, and it was a radical idea of patient power, and it overturns the traditional paternalistic nature of healthcare. And the problem is that it seemed like a very good idea at the time, but now we are reaping the reward of that rotten fruit, because now they've decided that, well, patients really don't understand, and I've come close to that, where they don't think that you can make a competent decision. So now we're going to turn it over to artificial intelligence. Not even the doctors and nurses these days know how to treat a patient. It's only science and artificial intelligence and the agenda that matters. In 1980 was the Diagnostic Statistical Manual, the DSM, and it includes autism for the first time. And this is the model that also has like 10 different ways that that doctors can get paid for killing a person. In 1980, autism cases increased to 1 in 10,000 people. In 1987, there is a spike in the autism spectrum, and advocates behavior therapy. So then, this was really the start of, okay, if you have any type of disease, especially if it's chronic, we are going to advocate behavioral therapy. It's all in your mind. It's your fault. You're a dirty, rotten fragile, flawed human. And so we need to fix you. And this is the year that this philosophy or ideology first began. In 1991, autistic people required special education. In 1997, autism cases go to 1 in every 5,000 people. In 2003, the Autism Genome Project was completed with inconclusive findings. So they didn't find necessarily an autistic gene. In 2006, autism cases went to 1 in 150 people. In 2008, the autism gene was discovered. In 2009, autism cases went to 1 in 110 people. In 2019, autism cases went to 1 in 59 people. And it included 1,050 genes in the people with autism. In 2001, autism cases went to 1 in 44 people, but this is a lag. There is a lag time because this number is actually from 2018, but it was not reported until 2021. And these cases are going to go exponentially higher with the advent of COVID and the treatment for it. So now we're going to go into directed evolution or directing it and genetic mutagenesis. So I personally do not believe in evolution, but the powers that be do believe in it, and they are no longer interested in following genetic traits that kind of ebb and flow with these minor little adaptations. They want to control all biological life as if they are gods. Given the common mythologies around the world, it is very likely mankind, having responded to some metaphysical force, engaged in this type of experimentation on Earth, but was destroyed. It may explain some of the major civilizations of which there's nothing left except stone ruins. Today we have a hydra or a Methuselah of various data points that are operational under the umbrella of directed evolution and experimental evolution. These two disciplines, in collaboration with many others under One Health, are under the supervision of what I believe fallen angels for the purposes of corrupting the bloodlines of man, Adam. Therefore, they call their operation Adam. We must make a distinction between Adam of mankind and the uppercase Adam. God's name for man he formed is Adam, A-D-A-M, with a capital A and lowercase d-a-m. In today's Adam, all uppercase indicates to us that Adam is owned by its corporate designer and is a corporate citizen and subject to its engineer, which for God's creation, they're subject to the creator. This Adam, uppercase Adam, is subject to the corporation and the people who engineer it. Adam is fully patented and controlled by its corporate owner. So now we're going to start delving into some of the science. It gets a little bit difficult for if you're not scientifically inclined, but I'm going to do my best. So what is directed evolution? So it is a method used in protein engineering that mimics the process of natural selection to steer proteins of nucleic acids or your DNA towards a user-defined goal. And that's very important, a user-defined goal. This is not a natural, like what might happen, like if a chicken moved to Mexico and didn't need as many feathers. This is engineered by man. It is a synthetic process that harnesses the power of selection to evolve molecules, typically proteins, because we're mostly proteins, or nucleic acid or DNA, from large combinatorial libraries to have the desired protein or properties. SARS-CoV-2 is directed evolution at work. And I want to draw your attention to the graph here. So we're going to start over kind of to your left where it says the natural gene. And I think I'm going to have to switch glasses. Sorry about that. Just can't. It's little tiny font. There you go. Much better. So you hear a lot about diversity. So what the powers that be, their definition is this directed evolution, where for us, diversity just means, you know, there's different kinds of birds, there's different kinds of cats and dogs, that type of thing. So they go over here and we're going clockwise, and they go to their gene library where they have different mutations. And so then they say, oh, I like this mutation. So then they get the protein and they nurse it or they incubate it. And there's a protein library. And they say, okay, they've kicked the tires on all this different proteins. This is why we have so many different gene editing tools, is because they're kicking the tires to see what works more efficiently to do this directed evolution. So they go, okay, this particular protein and this particular protein really work, you know, these two red ones right here, but these guys don't really work very well for our intended purposes. So we're not going to select them. So this is like the selection process. And this is very similar to the Holocaust, where they selected different people for different jobs, that type of thing. And then what they do is they amplify that particular mutation and protein or gene or whatever that they're working on. And then they replicate it over here. This is the replication factory. In fact, they are turning your body into a bioreactor so that in case they're not around, once you get the platforms into your body, you will just be generating and replicating these on your own. It's a devious plan. And then they call this an improved gene. So they're actually destroying humanity, but they call it an improved gene. And so this kind of gives you an idea of the flow of directed evolution. So we're going to go on. Now, what is experimental evolution? It is the study of the evolutionary processes occurring in experimental populations in response to conditions imposed by the experimenter. Now we've got the pharmaceutical companies who are the experimenter, and we are all, the whole world's population, at their complete disposal for as an experimental population. We are in a huge concentration camp, and the Dr. Mengelas of the world are experimenting on us. This research approach is increasingly used to study adaptation, estimate evolution perimeters, and to test diverse evolutionary hypotheses. So they're not happy to just have those hypotheses in computers or in books or in drawings. They want to test it and experiment. And so that's what they're doing. And so SARS-CoV-2 is experimental evolution at work. These experiments involve changing a human into a synthetic entity, and I am going to be describing that in great detail. This is a metamorphosis process exploiting the metals in a person's body or through the introduction of additional metals. And recently, I don't know, maybe for the last year, a lot of people contact me and they say, should I take nano silver? Should I take gold? What about copper? And all of these traditionally throughout history have been very wonderful remedies and used for healing people of different afflictions. unfortunately, as we're in this directed evolutionary process, these metals can be exploited and turned against us. And that's why, at least at this time, until we really understand the mechanisms a little bit better, that we might want to refrain from consuming a large quantity or using these where we might have freely used them in the past. So robotics are primarily metal, but now include a fusion of biological life and metal. You're going to see that increasingly over the days. So the current experiments use your own enzymes or synthetic or artificially implanted enzymes to achieve the experimenter's desired objectives. One of these enzymes that can be manipulated is called, now I'm going to try really hard with these words, metalloproteinase, which is any protease enzyme whose activity mechanism involves a metal. Any of several proteinases have a metal atom in their active center. So basically, you've got a metal plus a protein plus a conversion by an enzyme. And I did, even though they're abstract and they're not exact pictures, I wanted to even, they're computer models, but I wanted to give you an idea, a visual of what is happening inside of our body. So I am using these. Are these the actual thing? No, they're not, but they're a model of showing how this process is occurring in your body. So does anybody have any questions?
There are a couple of questions, Celeste. Let's go with the questions that we have. One person is wanting to know, is Operation Adam a subset of Operation Dionysius?
I don't know what Operation Dionysius is, so I don't know the answer to that.
Okay, another comment from somebody here says, just to say Adam is featured in the video game series BioShock as a building block of genetic modification, pretty much exactly as you describe. And another comment here, let me see. The newest injection says to be proteins and very safe, as it is more like the old type of injections. Why do they point out the proteins? I'm not sure if that's directly relevant, possibly not.
So my comment to that would be, if you believe them, I feel sorry for you, because they're not really, this is a big project. They've spent billions of dollars on it, and they're not going to tell you, they're going to make it sound very good. And case in point, if go and watch my Dr. Evil toolkit this week, and in one hand he says, this is benevolent, it's for your good, and then he proceeds to say how it's going to harm you and kill you and all the different ways it will kill you. So, you know, we can't trust what we're being told.
Okay, some questions about autism and mental retardation. Is it the same thing? I don't know whether you want to address that.
I don't think autism and mental retardation are the same.
Okay, I think that sums up the questions that we have just at the moment, as far as we are right now, Celeste.
Okay, sounds great. I just don't want to get into deeper waters and then have you like all confused. It's easier to answer your questions kind of as we go than to get into the deep water and have you like floundering around and not knowing. So, okay, so basically we have God's Adam at war with Adam, or Adam, this new cyborg Adam at war with humanity is basically what it is. So the Bible mentions 2 Adams. There are no other Adams mentioned in Scripture. The first Adam was the first man created from the dust of the earth. in the image of God, and breathe with a breath breathed into him by God. The second Adam is Jesus Christ, the Son of God, who died to save man from his sins. The first uniquely named Adam belonged to mankind exclusively, the name. The second name, Adam, is reserved for Jesus Christ alone. Today, we have an engineered, synthetic, evolutionary, cybernetic Adam, and that's capitalized. This evolutionary Adam can take one of two forms: a manufactured synthetic entity, humanoid in appearance, with no soul, but is a habitation for evil, or two, a directed assault against your body and my body, resulting from the insertion of soft robotics that slowly transforms you and I from a human into a hybrid, and then to a synthetic shell to be used as a host. Now we're going to get into Operation Adam. How comprehensive and widespread is this evolutionary attack against all biological life, specifically against humans? To learn the answer, we must investigate the acronym ADAM to glean some insights and look at the lineage of the synthetic Adam. As you view the following acronyms and lineage lists below, note the proclivity to interfere with the human reproduction system. So the first, we've got the arrestee. I'm not going to say Adam, you can see it for yourself. It gets a bit redundant. So it's the arrestee drug abuse monitoring. We definitely have that in play right now. The various disciplines involved are psychiatry, law enforcement, and government. Now, there's a mixed case, and this is the only mixed case, but I did put it because it is analysis data model that is for health and care and drugs. And that is the only reason that I put this mixed case. And so this might indicate to us this particular analysis data model might be watching the transformation of the original Adam into this synthetic cyborg Adam. Then we have the androgen decline in aging males. This affects the testosterone, hypogonadism, and aging. So what is an androgen? In case you don't know, it's a group of hormones that play a role in male traits and reproductive activity. It is present in both males and females. Then there's another, now some of these words, when they get to the chemicals, I'm not even going to slaughter them. You can see them, they're there for you can use them how you wish later on, but it's Adam. The short version is Adam, and it's used in chemistry. And what is it? This novel Adam analog has been synthesized, pay attention to words, and enhanced, so this is artificial, potency as an anti-HIV agent. This drug is given due to the manifestation of the HIV genetic material inserted to you via the spike protein weapon. I'll let you think about that for a minute. So they're going to use a synthesized and enhanced anti-HIV agent because they inserted this genetic material into the population. Now this next one is very interesting. It's amniotic deformity, adhesion, and mutilation. I mean, instantly when I think of the word mutilation, I think of the cattle mutilations. The disciplines involved in this are health, body, and healthcare. And so what is this? First and foremost, I want you to notice that they do call these mutilations. Next, I want you to see that it includes craniofacial defects and limb anomalies, especially from amniotic deformity inside the womb, adhesions, and mutilations from the Adam complex. This Adam complex has facial abnormalities that can comprise of clefts and distortion and dislocation of craniofacial structures, such as elongated skulls. And I say this because, well, I'll finish this and then I'll go back to the elongated skulls. Limbs show various combinations of amputation, secondary syndactyly, or web fingers and toes, and constriction. Now back to the elongated skulls. So about 2 months ago in the journals, I noticed that they did this one study, and it was talking about putting a swab up a nose, up to the blood-brain barrier, and that they had certain genetic material on the end of that swab. And what that genetic material did was seed the skull. And they could, in fact, change the shape of the skull by this seeded genetic material. I believe that they had this technology maybe at the time of the Egyptians, maybe down in Peru, maybe some of the other places that had elongated skulls, and that so that they had this technology, and they simply took a swab and up the nose to the blood-brain barrier with this seeded genetic material. And it will be more difficult to change the shape of an adult skull, but it would not be so difficult to change the shape of a child's skull, an infant skull, or in the womb. There's the Amsterdam duration of antiretroviral medication. What disciplines does it include? Health, body, and healthcare overall. What is this? It's an antiretroviral drug which inhibits the production of retroviruses, and those are comprised mostly of DNA rather than RNA. And the best known group is HIV, which was inserted into those who acquire the spike protein weapon either from the environment or pharmaceutical editing tools. Then once again, some of these can be redundant, but you'll see patterns, and it's important that you pick these patterns up. Androgen deficiency in the aging male, testosterone deficiency, androgen, and I just highlighted, I didn't put the whole thing that I was looking at, but it has to do with male reproduction. Although, as I mentioned, it can be in females, but it's specific to male reproduction. Then there's the aneurysm detection and management. And that has to do with the disciplines that study aneurysms, aortic, and abdominal. And what is this? It's a surveillance and management of unruptured intracranial aneurysms caused by the spike protein weapon. It deals with blood vessels in the brain, and we will be talking about it a little bit more thoroughly a little bit later. There's angular distribution auger microscopy that deals with chemistry, technology disciplines. And what is this? This atom is useful for direct imaging of the interfacial structures and investigation of electron-solid interactions in the physical and biological sciences and engineering. So they are imaging as they change your facial structure and what goes on even in between the structure, the physical structure of like your brain, in the biological sciences as they re-engineer your body and your brain. Kind of scary. Then there's the animated dissection of anatomy for medicine, and what disciplines are involved in that: technology, anatomy, and dissection. This Adam software is a multimedia interface for the universal integration of medical knowledge. And they're not talking about the integration of in the computer. They're talking about this is a universal integration of how they're going to change your body. So it's and they're kind of dissecting you. And so if you notice in the journals, it'll say, okay, we're gonna dissect the heart, and we're gonna make a synthetic heart. We're gonna dissect the circulatory system. We're gonna make an artificial circulatory system. We're gonna dissect the brain. We're working on getting an artificial brain. And all of this technology is moving towards putting a totally biomimicry system into a synth or a synthetic humanoid. Then there's Adam, a disintegrin, and metalloproteinase, and that's in biomedical, bioscience, and biochemistry. So we're going to continue. Adam is also, I'm not going to, like I said, these chemical words, I'm not going to say them, but you can see it. What is this? It is rattlesnake venom. And I want you to remember that about 2 years ago, when everything came onto the scene, I said at the time that we start seeing snake venom or serpent venom in pharmaceutical products, we are getting close to what I believe is the mark, or a very, if it's not the mark, it is a very lethal time for humanity. So here we find rattlesnake venom, and there are other studies. You can find articles about, if you look up rattlesnake venom and COVID, but we will be going into it. And there's also other venoms. They're using the most, since World War II, they've been trying to get the most deadly venoms on Earth. What is that? Zebrafish and some other venoms, and get them into the human body. So it is a generic protein in the family of whatever, in the superfamily of metalloprotease, which include Adam, a disintegrin and metalloprotease, and the Adam TS, a disintegrin and metalloprotease with a thrombosidin motif. And thrombosidin, if I'm not mistaken, is your either ability to clot or your inability to clot. It influences that. So the venom this enzyme adamolysin is the venom of the eastern rattlesnake. The venom contains a compound called crotalase that clots the fibrogen, ultimately reducing the platelet count and rupturing red blood cells. Another venom component is a neuropeptide that can cause cardiac arrest. You definitely do not want this in your body. The venom causes, if you got bit by an eastern rattlesnake, it would cause bleeding, swelling, discoloration, extreme pain, tissue necrosis, and have low blood pressure. So when you see the serpent venom, beware that the serpent of old is at work. So here's the adaptation and anticipation model. So they're adapting these humans and all biological life. It's not just humans, but they have a special affinity for humans, and they're anticipating with great expectation, what will we come up with? And this one is particularly in the discipline of neurology, which is our brain and nervous system, basically. The next Adam is adaptation and behavioral experiments. I didn't go into that too much. Adaptive moment estimation, that has to do with optics and photonics. And what is it? This is tracks decay. This Adam is a method for computer optimization, quantum computing, solving linear systems with noisy data. The bottom line, a decay or degeneration process inside of you has been initiated and is being tracked by quantum computing. And that's why we're shortly going to 5G and then 6G in a way. The next is, I don't really understand this one, but it's Adduct Detection, I'm not even going to read it. It's in the disciplines are therapy, health, and body. What is it? It's an analytical procedure for detection and quantification of DNA adducts, which means drawing towards the center of the human tissue and cell-free protein translation and cell labeling assay, for those of you that want to know that. Then there's adhesive dermally applied microarray. Most of you are familiar with that one. It's involved with the discipline of neurology. This atom consists of the titanium microneedle array attached to an adhesive backing seated with a retainer ring and an applicator, and it began with DARPA. That's that one with the death by 1,000 needles. So here we go into Adam, a disintegrin and metalloprotease. This is health, body, and healthcare. What is this? The first disintegrin protein is from snake venom. Identification of mammalian membranes anchored in the metalloprotease and disintegrin is implicated in fertilization. So when they use this venom enzyme, it throws off fertilization, or it can increase fertilization if, let's say, they want to increase the fertilization of the parasites that have been implanted. I was shocked to find out that the studies have been going on for 3 decades. That's 30 years, folks. And have been identified additional members of these anchors and several biochemical mechanisms regulating their expression and the activity in the cell. Does the disintegrin anchor to your cells? Yes. And this influences female fertilization. Where most of this was targeted at the male, this particular snake venom was aimed at female fertilization. So what I found were 77 Adam, capitalized Adam acronyms, some applicable, some not, but I have provided only a sampling of them. And notice that 77 is a biblical number, so I do not think it's by coincidence or a mistake. So are there any questions?
Yep, there is 1 or 2 questions here, Celeste. Let's get to the questions that we have here. Somebody wants to know about, I know this is a little bit earlier, but they want to know, did autism actually spread through vaccines? How did it spread?
This really shows, I can't answer that because I didn't have time to study this. I had to get this done because I actually have a different webinar that I was supposed to be working on. But this really shows a direct link between transplantation and the blood and autism. And so, we can get blood in different ways. Don't forget, Bill Gates put blood in rice, golden rice. It's been in tomatoes. It's now used as aborted babies are found in as natural food flavorings. So we're getting inundated by blood, genetic blood from humans. And so I think, and then we see this, steep, it's just, who knows, how many, we're on the downhill as far as the frequency of autism. And I think it's directly related to the blood. And there's power in the blood. Sacrifice has always been about the blood. I think really, I know that it might have something to do with vaccination, whether they put some blood in the vaccination products, I don't know. Like I said, I didn't study it, but I know that there's a direct correlation between the autism and the blood. So,
did you mean transfusion? You said transplanting, but did you mean transfusion there, or did you mean transplant?
Well, there's blood transfusions. We can get it different ways. It's transplanted our blood, genetic blood materials are put in our food supply. It's They've said that it's in the geoengineering. I've never seen a study on it, but there are people that have studied the fallout from geoengineering and say that there are blood elements in there. So I think we're going to see it exponentially rise in light of the COVID and the requirement of testing and this whole emphasis on precision medicine, which requires a blood sacrifice from you. And then they can genetically edit that and even send it, as we learned with Dr. Evil's toolkit, send that remotely to you and edit you and impact your body and your brain. So we live in a very serious time. They don't even have to come in contact with you. You don't even have to have eaten it anymore. They can send it remotely through frequency to your body.
So another question here about a drug that you mentioned. I'm not quite sure the pronunciation of this, but it's alkendierymethane drug that will be given due to the manifestation of the HIV genetic material inserted via the spike weapon. How will they give this medicine? What is the medicine called, and what excuses or lies will they tell in order for people to submit to the drug? Somebody wants to know.
Well, so if you go to your doctor, whatever the symptoms are for HIV, which are many, you go to your doctor and he runs some blood tests. Let's say he runs a panel on you and he comes back and he says, you've got HIV. That causes panic in people. And so they're instantly going to offer you, some type of pharmaceutical treatment plan. They're not going to offer you like a natural thing. And then how did you get HIV if it wasn't from this spike protein, which did have HIV as part of its original construct? I mean, and you didn't sleep around or whatever. The only way that you could have gotten this HIV was through this spike protein that had this HIV genetic material in it.
Okay, now somebody else wants to know, why would they want to change your skull shape? Why would anybody want to do that?
I just saw this for the first time 2 months ago in the journals. So I think that it's just now coming into the scientific literature. I don't really know the answer to that. I could speculate. People have had a fascination with elongated skulls. The ancients did. They used to think that they were elite. Maybe they thought that it was beautiful. Maybe they thought they were more intelligent. Some people did use the headboard treatment on infants, but many of those infants that have the elongated skulls do not show indication of the headboard. So this might answer the question. For those that believe in panspermia and a reptilian race on Earth, and I know some of us believe our politicians are reptilian, are they trying to take humans created in the divine image of God and turn them into reptiles, which in my biblical worldview would be Satan, the serpent of old? That's what I see happening, and why they would want to change the skull. They want to change everything, but there's certain things that are more enticing, like the blood, the skull, that type of thing.
Okay, and there's somebody here worried about this snake venom treatment. Person says that they were administered some kind of snake venom by their chiropractor, and worried about what other effects there could be from the snake venom going forward.
So I highly recommend if you put anything in your mouth or consume it or even put it on your skin, because your skin is your largest organ on your body, know the ingredients, do your homework, research it. Don't take anyone's word for it. You may or may not find information out there just because, like doctors or nurses, or if you go to your grocery store, the synthetic biology. I mean, the grocery, you ask your grocery clerk, is this a tomato? If you ask this farmer's market vendor, is this a tomato? You know, what's in this tomato? They're not gonna know. So it's incumbent on you, depending on your level of being comfortable, do your own homework and say, I want to know ingredients. I want to know any adulterants. I want to know specifics. Ask for, if it's a pharmaceutical, ask for the professional insert, demand it. And if they can't give you a professional insert, do not take it. Do not take it. And then be aware that the technology today is being used, exploited, and manipulated. So what may be safe for you right now for a treatment, maybe down the road, they flip on the 6G and it does something entirely different into your body. So you're really going to have to be careful. And we don't have all the answers. I'm just now getting to understand some of these things. I didn't even know 6 months ago, I didn't even know about directed evolution or such a thing existed, or experimental evolution. So all I can say is I thank the good Lord that we found out. So now we can be more careful.
Okay. There's quite a lot of, you've raised quite a lot of questions about the snake venom issue. Somebody else wants to know, when snake venom ruptures the red blood cells, does this affect the uptake of oxygen into the body, causing breathing issues for people?
So we are going to be talking about the snake venom a little bit later. So let's hold the snakes for just a little bit. We're not done. We're like about a third of the way through, and we will be diving in, and maybe your questions will be answered.
Okay, and one final question for now, Celeste. Is this going to be available as a PDF so people can look up your links?
Yes. Yes.
Okay, that's it for now.
It might take me a day, depending on my interview schedule, but yeah, it is. Okay, so now the next thing is the lineage of Adam. The biblical Adam the first and Adam the second, there was proof of genealogy. It's critical to authenticate or verify the lineage. And the graph over on the right-hand side shows you the lineage of Adam and Eve and some of their offspring. I didn't go all the way just to make it easy on you guys. And so God sets the rules, and it's necessary for the serpentine counterfeit to provide a lineage for the synthetic evolutionary cybernetic Adam. So that's why the elite have to tell us first, because God told his prophets first. So the rules are set by God, and then the counterfeiter comes in and he has to do the same thing. So we have this lineage of Adam. And I will tell you up front, let's see. Okay, well, I'm going to tell you right now because I don't want to forget. It might be in here, it might not. There are 33 Adam iterations. So there, and that, as you know, is an occultic number. And we're not going to go into the occultism in this presentation, but the next time I do a webinar on Adam, we will be discussing and diving into all the occultism that's embedded in this Operation Adam. But for today, we've just got our hands full just trying to understand the very basics of the biology and the spiritual and that type of thing. So several of the disintegrin and metalloprotease or Adam family members are expressed in the male reproductive organs such as the testes and the sperm. The reproductive atoms are phylogenetically divided, and phylogenetics is the phylocode, which I talk about quite often, using the clade system that replaced the lineage system of life classification. Well, what's so wrong with that? If you haven't heard my spiel, I'll just do it in a couple words here. So basically, a human used to be a human. The dogs were in the dog family. The cats were in the cat family. Well, the clade system, they worked on it for 10 years. It's evolutionary. And they decided that humans needed to be in the clade of arthropods, birds, and dinosaurs. So those are our family members, according to them, in this new evolutionary world. So it's a very messed up system. I won't say anymore. So there are 3 distinct major groups. I'm not going to read them off. You can see them right there. There's the first group of atoms. It's not atoms, it's atoms. And the second group and a third group. And they're decreasing in the number, and you'll see why in just a minute. Okay, so this is this shows genetically modified knockout mice. And first I'm going to describe a knockout mouse is a laboratory mouse in which researchers have inactivated or knocked out an existing gene by replacing it or disrupting it with an artificial piece of DNA. So it's been gene edited. Now I'm going to go up to my first bullet point. Previous mouse knockout studies indicate that Adam 1, 2, and 3 have intricate expressional relationships playing critical roles in fertilization. And it shows how this is done. And it's done through the blastocytes. And then the last, it ends up being a genetically engineered mouse. In the present study, a comprehensive analysis took place on the previously unexplored second group of atoms. They found that all 3 atoms are made as precursors in the testes, and that atom 5 and 32 are located on the surface of the sperm. Researchers then used the sperm to make genetically edited knockout sperm. So these are artificial sperm, you guys. These are genetically modified knockout sperm. Taken together, their results suggest a novel expressional relationship of atom 5, 7, 2, and 3, which play critical roles in fertilization. So, there's several problems with this. One, the fertilization of the parasites, the fertilization as we genetically edit humanity. What type of creatures are we creating? What abominations? Or are we sterilizing? So Adam 1, fertilin, is a subunit of an integral sperm membrane. It's a protein or an assembly of proteins that is permanently attached to a biological membrane. Sperm egg fusion, and this was interesting, virus-induced cell-to-cell fusion interferes or interacts with the maturing of the sperm. Unique mechanisms for modifying integral proteins in the sperm plasma membrane. There is an awful lot in the journals right now about membranes, and what they're trying to do is get rid of all the boundaries, just like with the borders. Think of your cell or the sperm as the borders, and they want this free flow. They don't want a border. They just don't want any barriers. People come, people go, whatever. Everything except intellectual property, of course, that you can get in trouble for. But so it's the same way. They don't want the sperm to have this, to be a sperm. They want to be able to inject things that they want into it and affect or influence it to do certain things. Like in certain studies that I'm seeing, they have cut off genetically the tail of the sperm so that it cannot swim. So Adam 1 is a heterodimeric glycoprotein, which is a sugar-coated protein, and it plays an important role in sperm-egg interactions. And I try and give you, here's a glycoprotein right here, and it looks much like a carbohydrate. Now, remember, they don't want us to have any carbohydrates, and they don't really want us to have any proteins except modified in the laboratory. So they want us to have this totally synthetic world. And so does it surprise you that they are doing this? So what is a heterodimer? It's a molecule composed of paired proteins with some amino acid sequence variations. Pseudogenes are thought to be non-functional segments of DNA that resemble functional genes, but this is not the case. Scientists used to refer to this as junk DNA, but it's not. Every bit of genetic material has a purpose. Just like in the old days, they thought that space was a vacuum. They now know that it's got plasma, dust, all sorts of things. It's not really a vacuum. Most arise as a superfluous copies of functional genes, either directly by DNA duplication or indirectly by reverse transcription of an mRNA transcript. And then I'm not going to bore you with all of this for doctors and nurses. It's there for you if you want. And this just, I wanted to show you the difference between a human and a mouse, because we've got these humanized mice and they're getting edited into our body. So I just wanted you to see the difference. This is the mouse, this is the human. So Adam 2 is a subunit of the integral sperm membrane of the heterodimeric glycoprotein called fertilin, which plays an important role in the sperm-egg interaction. So it acts much like Adam 1. Adam 3 is a disintegrin, and a metalloprotease 3 has not been found in humans. But is this going to change with all the gene editing and the humanized mice being inserted into humans? I don't know the answer to that. Adam 3 belongs to a family of peptidase proteins referred to as Adam. Many of these are solely found in the spermogenic cells at the front of the spermazoa head. The membrane protein is critical for crucial steps in fertilization, such as the migration of sperm through the uterus to the oviduct, as well as binding to the zone of the pellucida. Inactivation of Adam 3 causes male infertility. Numerous studies have detailed that Adam 3 and null mice, which are knockout mice, the spermazoa fail to migrate through the uterotubal junction. Furthermore, the knockout of the closely related Adam 1A gene in mice, an estrogen receptor not found in mature spermazoa, causes loss of surface Adam 3 and decreased zona pellucida binding, basically the connection. Other studies have shown Adam 3 mouse spermazoa fail to show sperm to sperm aggregation where they collect and they all go together, although the significance of this is still unknown. This is a picture, this is Adam 4 in what you would like a model, and these are the Adam 4 antibodies. This is Adam 5. Some of these are either redundant or there is not a lot of information on it. This is probably a pseudogene, which means it's not a real gene. Adam 7 is a transmembrane. Remember, I told you that they're really interested in making things fluid. They don't want these solid barriers. They want they want to be able to put things in and take things out at will or on command. So things are going to a transmembrane. They want everything to be like a transmembrane protein. And this transmembrane protein originally was important for the maturation of sperm cells. This gene encodes a member of the Adam family of the zinc protease. These transmembrane proteins play roles in multiple processes, including cell signaling, adhesion, which is the sperm and egg fusion, and the migration. Mutations in this gene may also be involved in the progression of melanoma. Adam 8 is a protein encoded by this gene, may be involved in cell adhesion, coming together of cells during a degeneration of neurons. Adam 9, protein encoded in this gene interacts with the SH3 domain containing proteins. And this has to do, so proliferation and survival, immortalization. Is this where we get, is Adam 9 where we get these immortalized cells that live forever in the laboratory that can just be experimented on? Cell adhesion is the coming together of the cells, and the angiogenesis and metastasize. We're going to be talking about angiogenesis in a few minutes. Growth factor, which is really important in all of our lives, especially for children, but even as adults, it will make the difference between whether your obesity factor. Adam 9 is, I just want you to remember Adam 9 until our next presentation, because this is having to do with the ectodomain shedding, and it's also called angiogenesis, which I just said we would talk about. I'm not going to go into it's a protein that blocks the function of key molecules that are required for complete cell division. It's involved in TPA-induced ectodomain shedding of membrane anchored. And then ectodomain shedding is a process that is highly regulated by specific, I always can do antagonists, but agonists and intercellular signaling pathways, which it's really important to have that signaling. Now, this is where it gets really kind of interesting. So moreover, 2% of the cell surface proteins are released from the surface by ectodomain shedding, indicating cells selectively shed their protein ectodomains. And there's a reason. We're going to go into that in just a sec. But just remember, 2% of the cell surface sheds its proteins. Now, remember the ectodomain shedding, as I'm going to explain the dangers of ectodomain shedding a little bit later. And it's also heparin binding. Adam 10 is a sheddase and has broad specificity for peptide hydrolysis reactions. A hydrolysis reaction in living organisms are performed with the help of catalysts by a class of enzymes known as hydrolase. A biochemical reaction that breaks down polymers, which is the binding, such as proteins, and peptides are the bonds between amino acid. It's kind of like peptides are like the glue. Nucleotides, complex sugars, and starch and fats are catalyzed by this enzyme. It's really important. What is a sheddase? A sheddase are membrane-bound enzymes that cleave the extracellular portions of the transmembrane proteins, releasing a soluble ectodomain from the cell surface. And I give you a picture of that process right there. Many Sheddings are members of the Adam family or the base proteins. Adam 11 gene represents a candidate tumor suppressor gene for human breast cancer based on its location with a minimal region of chromosome 17q21 previously defined by a tumor deletion mapping. Adam 12 is a metalloprotease that binds insulin growth factor binding protein, and it appears to be an early Down syndrome marker. I also am wondering if it also is not involved with both the growth factor and insulin if it hiccups. I didn't go into some atoms, but I just left them as place markers because I think it's really important to realize whether they're redundant or if there's something new coming down the line. We need to keep the placeholders that there are 33 iterations of Adam at this point. Adam 15, through its disintegrin-like domain, this protein specifically interacts with the integrin beta chain, beta 3. It interacts with SRC family protein, the tyranase kinase, in a, I'm not even going to go there, suggesting that this protein may function in a cell-to-cell adhesion as well as cellular signaling. Adam 15 is classically significant in cancer and arthritis. This is a picture of Adam, well, I think we got the picture on the wrong one because this is Adam 17 right here, but it says Adam 16. So just think about it for the next one. Got on the wrong page. Is understood to be involved in the processing of tumor necrosis factor alpha at the surface of the cell and from within the intercellular membranes of the trans-Golgi network. It also has a role in shedding the L-selectin, a cellular adhesion molecule. Once again, that's, you know, sticking together. Adam 17 may facilitate entry of the SARS-CoV-2 virus, possibly by enabling fusion of virus particles to the cytoplasmic membrane. It has a similar ACE2 cleavage activity as the TMPRSS2. By forming soluble ACE3, Adam 17 may also have a protective effect of blocking circulating SARS-CoV-2 virus particles. So it may get into the cell, the cytoplasmic membrane, into the cell, but then it through the ACE3, it doesn't allow it to circulate. So whether that's important or not, I'm not sure. So the Shedding's activity may contribute to COVID-19 inflammation by the cleavage of the TNF alpha and the interleukin 6 receptor. And then somehow we got, oh, Adam 18 didn't have anything. Okay, Adam 19. The protein encoded by this gene is a sperm surface protein and is synonymous with Adam 27. The member, this member is a type of transmembrane protein and serves as a marker for dendritic cell differentiation. Dendritic kind of looks like trees. It has also been demonstrated to be active in metalloproteinase, which may be involved in normal physiological and pathological processes such as cell migration, cell adhesion, sticking to sticking, cell to cell, the cell matrix, and the signal transduction. I did put for your viewing pleasure, if you're having insomnia one night, a video on transmembrane extracellular matrix, the inner cell of life, the inner life of cells, which is actually pretty interesting. Now we go on to Adam 20, exclusively expressed in the testes, and it's synonymous. Adam 21 is synonymous with Adam 31. Adam 22, this gene is highly expressed in the brain and may function as an integrin ligand in the brain. Adam 23, the gene is highly expressed in the brain and may function as an integrin ligand in the brain. Adam 24, the plasma membrane protease present on mature sperm that may be involved in sperm function during maturation and or fertilization. And Adam 25 appears to be mouse, but it's found on the surface membrane of the sperm and may be involved in spermatogenesis. And I'm not sure if that is like a synthetic sperm where they're altering the genesis of the sperm. I don't know the answer to that question. And fertilization. Adam 26 is also on a mouse, but it's located on the sperm surface. And the reason I mention these is because of these humanized mice and knockout mice. Adam 27, sperm surface, again in the testes. Adam 28, the protein encoded by this gene is a lymphocyte expressed Adam protein. Adam 29, now this is a bizarre one, is a human Adam protein, 29 protein that is raised in insect cells. Yes, and then purified. Yay, yay. And then in a multi-step protein-specific process to ensure crystallization grade. Now that does not make me feel very comfortable at all. It's a state-of-the-art algorithm used for plasmid design or gene synthesis. A plasmid is a small, often circular DNA molecule found in bacteria and other cells. Plasmids are separate from the bacterial chromosome and replicate independent of it. This particular atom is German made, and it may be involved in the spermatogenesis and fertilization. And it seems to be a non-catalytic metalloprotease-like protein. Then there's Adam 30, which is human. It's in the human domain. It's imported, but they don't, and they say it's Homo sapien, but they don't say where it's imported from. That doesn't make me feel necessarily very comfortable, because God only knows where it's imported from and or who or what. And then there's Adam 31 and 32. And then we've got the last one, which is Adam 33, which is a protein, is a type 1 transmembrane protein implicated in asthma and bronchial hyperresponsiveness. Now, I want you to think about this in relationship to the SARS-CoV-2. And it's a transmembrane protein, a type of integral membrane protein that spans the entirety of the cell membrane. Many transmembrane proteins function as gateways to permit the transport of specific substances to cross across the membrane. Remember, they want that free flow in and out at their command. They frequently undergo significant conformational changes to move a substance through the membrane. And I am providing you a couple videos on transmembrane proteins that I found interesting. And remember that there are 33 Adam iterations, and 33 is also associated with occultic numerology and Luciferian worship. So do we have any questions?
We're all speechless, Celeste, but we do have some questions here.
Okay. I'm speechless too.
Well, yes, we need our advanced techie heads on to follow some of this. So let's see. somebody wants to know, how do we prevent from being gene edited?
Drink pure water. If you can get well water, that's best. Grow your own food. Be very careful anything in the commercial market can and will, and is designed to edit you at this point. So limit anything that you buy from the commercial market, especially if it's anything that you consume or put on. But to be honest, there's no way. They've aerosolized the platforms. They put it in the water system. They put it in our food. It's on everything that we've come in contact. They can paint it on us remotely. So at this point in time, you just have to just say, we're going to cry out to God and call upon him to protect us. And if we are injured in any way, to heal us. And if you do have a biblical worldview, there is a promise in scripture that says that you are a new creation in Jesus Christ. And so that's every moment of every day. And so he is warring for you. And, but all we can do is take precautions to the best of our ability and realize that this is an impossible situation, but this is another thing that the Bible said to us. It was written over 2,000 years ago, and it said, Jesus said that if the days were not shortened, there would be no flesh left alive. And we are actually facing that right this minute.
Okay. Now, somebody wants to know, did you study any other diseases other than autism?
No, I had like a week to put this together because I was working on a different webinar altogether, but I felt that this was so important to get out that we just, I've been burning the midnight oil, didn't have a holiday to try and get you this information. So I didn't have time to study other diseases.
Okay, now there's a couple of questions about these Adam iterations. Somebody actually says, and this I kind of track with this one. Somebody said, "What about first Adam 1 and 2? They were pretty good guys." And I thought, yeah, I like that too. But there's a question about what are all these Adam iterations, Adam 33 or 29 or whatever. Can you explain what these Adam numbers that you're talking about are, so that somebody could understand that?
You're so funny, John. Like I had somebody say, could you please put it in a nutshell? Like everything that you know. It's like, no. So if I had to say, these are vulnerability. These are areas in our body that evil powers are focusing on to disrupt and evolve humanity into a hybrid, into a synthetic being to be used as a host. Simply, these are the breaking points, and you can see. I mean, a lot of them have to do with reproduction. Are we going to be able to reproduce? If we are able to reproduce, what are we going to reproduce? These types of things. Are we going to be sterile? That type of thing. But then, as you saw, they weaved in and out this different diseases, cancer and HIV and other things. So you can clearly see that they're weaving it. It's like they're weaving this evil tapestry, this dark tapestry of darkness.
So is it fair to say these are gene mutations, or what, are they a complete DNA code, or are they just a part of it that's been modified, Celeste?
Well, they haven't necessarily been modified, but these are the areas that they're going after to be modified. So that's why. So like if let's say you live in your house, where let's say if a SWAT team came where would your house be the most vulnerable? Would it be the windows? Could they, crack down your door? Do you live in a very secure dwelling where, you've got a security system and, um, defense, mercenaries that come out and meet the meet the SWAT team? So basically think of each one of these atoms like your house, and the evil powers are trying to break into your house. That SWAT team is trying to break into your house, and in some cases they're successful. So they were not originally evil, but they are being edited and now are becoming evolutionary tools to change
Actually, it looks
humanity.
like you can go online and buy these proteins. So you can buy Adam 29, or you can buy, I guess you can buy any of them, and presumably use them in your human experiments at home. Would that be
That's called the DIY. And
DIY?
that's another thing that Dr. Evil was very worried about, was the DIY community. But let me tell you, if you are interested in joining the DIY biological directed evolutionary community, the CIA and other military has infiltrated to steer you in the right direction. Let's just say. We just learned that from Dr. James Giordano himself. So,
Well, if I ever think of setting up an experimental lab, I will ask for more advice, Celeste. But right now, I think I'm staying away from that.
yeah, me too. Okay, moving right along. So we're going to talk about Adam 10 for a few minutes, and then Adam 17, and then we'll conclude. And then in the next presentation, I will go into the occultism surrounding this whole Adam 10 operation, or Adam operation. So Adam 10 is a disintegrin and metalloproteinase domain containing protein 10, also known as Adam 10. I'm not going to read all that. It's encoded. What is important to note about Adam 10 is that the sheddase is a potent modulator of prion disease. And remember that genetically modified prion disease is what keeps Dr. James Giordano up at night. So if he's worried about it, we best be worried about it. And it's coming to us in 2022. A prion protein is highly expressed in the nervous system and is critically involved in prion diseases where it misfolds into a pathogen, which is what I'm talking about this. But it's even worse because it's going to be a genetically modified prion, which, yikes. Moreover, it has been suggested as a receptor mediating the neurotoxicity in common neurodegenerative proteinopathies such as Alzheimer's disease, and it also has a role in breast cancer. Adam 10 plays a role in the modulation of the molecular mechanisms responsible for dendritic spine formation, maturation, and molecular organization. And dendritic kind of means tree. And consequently, an alteration of Adam 10 activity is strictly correlated to an onset of different types of synaptopathies, ranging from neurodevelopmental disorders to autism spectrum disorders to Alzheimer's to prion disease. And then, so I wanted to show you this. Remember all those genetically modified mosquitoes? They may have had a lot. Many of us were suspect about those little guys. They said they were a good deal, but I'm beginning to think not. So here they're biting you, and then the different things that happen inside the infection, and then it gets into your liver. Well, actually, I show the liver on the next page, but it goes through this whole process of parasites in their different stages. And then this shows the hours at 16 hours, at 24 hours, at 32 hours, at 48 hours, and then they're bursting out. This is your red blood cell. And then it talks about the invasion. I think we're going to talk about the invasion on the next slide. Maybe. Oh, no, not quite yet. So here's here's another view here. Here's the little guys, and then it actually goes into our liver, and the liver stores some of these parasites that are being introduced. So these parasites, this is terrifying, actually, can invade your cells in 30 seconds. That's for malaria, that's how fast this process takes. This is important to know given the DARPA parasite program, along with the associations with the Acantha amoeba and the SARS-CoV-2. Now I'm going to talk about the process. So how is this process? I showed you the diagram there for a few minutes, but let's just talk about the process, because a lot of you are asking about transmission. And so this is going to start to answer your question. It might not fully answer your question, but ears alert. So proteins on the sticky surface of malaria parasites help the invaders stick or adhere to the red blood cells. So you've got the invader and it's stuck, because it's sticky, like honey or molasses. And then once attached to the host blood cell, the parasites need to shed the sticky surface protein that would interfere entrance into the cell. I don't know if you've ever got into super glue or even honey. I mean, it's really hard to work once your hands are all gummed up. And so these invader needs to get rid of that sticky, so because its mission is to get into the center of that cell. So then the shed-eyes enzyme is required for the parasites to invade the cells. Without it, the parasites would die. The shed-eyes is stored in and released from the cellular compartments near the tip of the parasite. So let's say, here's my parasite, and it's at the tips of the parasite is where the shed-eyes would be. And once on the surface, the enzyme attaches a motor that shuttles it from, I'm not done with my parasite, from the front to the back, liberating the sticky. The sticky goes out. And once the proteins are removed, the parasite gains entrance into the red blood cell. The entire invasion lasts 30 seconds. And so then that sticky goes off elsewhere. And without the Adam metallopeptidase, forget it. The parasite would be ineffective at getting into the cell. What else do you need to know about Adam and shed-eyes? Members of the Adams family are cell surface proteins with a unique structure. A shed-eyes is a generic name for the Adam metallopeptidase. I think my tongue is getting tired of this word. Peptidase functions primarily to cleave membrane proteins at the cellular surface. Once cleaved, the shed-eyes release soluble ectodomains with an altered location and function. The single shed-eyes may shed a variety of substances. So you need to understand that a variety of substances can be shed, and multiple shed-eyes can cleave to the same substrate or the foundation, resulting in different consequences. So different shed-eyes can do different things. Let's say make your eyes brown and your hair gray and that type of thing. Not really. I'm just doing a kind of a tongue-in-cheek example. This gene encodes an Adam family member that cleaves to many proteins around it. Adam10 is a sheddase and a broad peptide with a hydrolysis reaction. Hydrolysis reactions in living organisms are performed with the help of a catalyst by a class of enzymes known as hydrolases. And we talked about that a little bit earlier. The biochemical reaction that break down polymers or the glue, such as proteins, which have the peptide bonds between the amino acids and nucleotides, complex sugars or starch, fats, and catalyzed by this class of enzymes. In biology, the cell processes are involved in the cell cycle, apoptosis, which is programmed cell death. That is usually good. So like if you have cancer, your cells are supposed to just die rather than spread the cancer. Unfortunately, in our environment, that doesn't necessarily happen. A DNA repair and energy metabolism. So it is very important. In biotechnology, they are involved in protein engineering, which is making Adam and this new synthetic humanoid cyborg, proteomics, protein analysis, gene functioning, therapeutics, remediation. This was weird. Remediation of insecticides. Is that why we're not allowed to have ivermectin and nerve agents? So, and now we're, Dr. James Borodano is talking about poisons and toxins in the payload. So that are going to come against us this year. So, so that this is Adam10 is involved with this. Additionally, the vast majority of textile enzymes are hydrolases that are used in various industries such as textile, food, pharmaceutical, and ethanol production. So then you wonder, okay, is that good for if it's in ethanol, is it good that it's going out into the air? Adam10 cleaves to ephrin and within the ephrin F complex and is formed between two cell surfaces. I don't really understand this particular system. When the ephrin is freed from the opposing cell, the entire ephrin complex is endocytosed or ingested. The shedding in trans has not been previously shown, but may well be involved in other shedding events. What is ephrin? Ephrins are the cell surface molecules attached by either the GPI anchor, which is the ephrin A family, or the single transmember domain, which is the ephrin B, and serve as ligands for the cell surface receptor tyrosine kinases, in which the ephrin A and B families. Like I said, I don't really kind of get this. I tried, but in a hurry. It's very laborious to try and figure this system out. In neurons, Adam10 is the most important enzyme. It is important because it processes the amyloid precursor protein. And the amyloid plaques are which gives many people, or it's hypothesized that it gives people like Alzheimer's, dementia, that type of thing. Adam10, along with Adam17, cleave to the ectodomain of the triggering receptor expressed on the myeloid cells 2, called the Trem2, to produce soluble Trem2 or S-Trem2, which has been proposed as a CSF and sera biomarker for neurodegeneration. So they're watching that as a biomarker with their imaging. Everything is imaging us right now, unfortunately. The Trem2 receptor is a transmember protein that is made of an extracellular region, also referred to as the ectodomain, the membrane traversing segment, and an intercellular component. The extracellular component of the TREM2 can bind to different ionic ligands, especially glycoproteins and lipids, like your hydrogel. It can silence inflammation. It allows for unrestrained replication, as in hydrogel. And a proposed active site for ADAM10 has been identified by sequence analysis and is identical to the enzymes in the snake venom metalloprotein domain family. So this is important to remember. The TREM2 receptor is a transmembrane protein that is made up of extracellular region referred to as the ectodomain, and the membrane. I thought we just did that. Maybe I did that twice. The extracellular component, yeah, I think I did it twice. So this gives you, here's the TREM2 right here. It'll be easier when you can just look at the diagrams close up and not have me talking. So what else do you need to know about Adam and the Sheddings? So the disintegrin, a family of snake venom, specifically the viper, anticoagulants that bind to the integrins of the surface of platelets and the cells, preventing their interaction with extracellular matrix and other cells. They inhibit tumor growth and angiogenesis. Angiogenesis is defined as the formation of new blood vessels, the formation and development, I just had the formation of new blood vessels, and also engineered synthetic blood and circulatory systems. Eventually, the reason they're playing with this angiogenesis is to put an artificial circulatory system in a synthetic humanoid. Also, any family of polypeptides isolated from venom that interact with the integrin proteins and inhibit the clumping of platelets. So what you're going to be seeing, or what is going to be in the journals that I look at, is that we are going to see rattlesnake venom being used in certain pharmaceuticals, and now also viper. So I think that's any questions?
Okay, Celeste, yes, we do have questions here. So let's go to the questions. So back to where we were, I think some of us are still trying to understand these Adam protein designations. So this person writes this. Thus far, I understand these Adam numbers as different substances patent and owned and controlled by its designer to be used to somehow insert, inject, inhale into humans. Is that correct? This person writes.
So they are parts of the human biological system that were a part of Adam lowercase. I'll put lowercase as distinguishing it from uppercase. But through editing, they're being changed into uppercase Adam as weapons formed against us and can be used against us. So like the analogy that I used with the SWAT team trying to find the vulnerabilities in the house, they're using these things. So everything is being mapped right now. So the strengths and the weaknesses are being mapped in every, like, fertilization and, you know, what makes us breathe and this and our brains and everything. And so they're just mapping, and then now they're kicking tires on different things, and we're going to see them with all these, you know, more variations of the pathogen coming out. We're going to see different pharmaceuticals being released, but it's all a giant experiment, and it is purposely to evolve man into this hybrid that is ineligible for salvation.
So there's a couple of questions here relating not only, well, partly relating to food, but also things that are spread into us via the upper atmosphere. So, a question people are asking, can these methodologies of altering people's genetic makeup be applied either through food or through things that are sprayed into the atmosphere?
So Dr. Evil, which I did that webinar on Monday, and that information was only 2 weeks old. And it's the first time he's gone public on some of the information. And he wasn't talking to the general public. He was talking to NATO partners. It's going to be this year. These will be deployed via aerosol and transdermally. So things that we touch. Now, he didn't elaborate. I mean, I know it goes through the skin, but is that through your lotion, or is that they're just going to put like smart dust everywhere, and when we touch something, then it goes into our skin? I don't know the answer. He did not go any deeper. He just said that this new toolkit that he has for editing is going to be aerosol and transdermal.
Okay, so of course, people are asking, are these platforms, or I don't even know if that's the right word, in organic produce? And another question, will any of these atoms be put into shots for children, for example?
So these are proteins, and so the war is against human proteins, and they are modifying the proteins. They can do it as Dr. James Giordano says, drugs, bugs, toxins, devices, and the devices do by frequency. So no longer does it have to be just, you went and got a platform at something that had hydrogel in it at the store. I can sit here right now and I could be inserted via frequency with whatever they wanted to do. It is much harder if we are a human, just a pure human. They could make our life pretty miserable. Just listen to anybody that's in the targeted individual community. But when you have the platform, they can deploy, they have so many different options that they can deploy toxins, bacteria, viruses, prions, parasites, explosives, anything into the human body. So we live in this very precarious time.
One of the questions relates to, but before I get to that question, so just to summarize, Celeste, what we are talking about here, what you are discussing here in this webinar today, in this expose, is you are discussing basically the state of the art of the methodology of trying to rewrite the human genome into a different kind of a human. That's what, am I right? Does that summarize what
Well,
we're
it wouldn't
doing
be a
today?
different kind of a human. It would be a humanoid, because it wouldn't be human anymore. It might
Well,
look like a human, but not.
okay, no, I take that point. But of course, the question then arises, which we've had this discussion before, but the question then arises, okay, at which point, at what is the threshold at which a person ceases to be human? You can mess with a person's genetics, and let's say you can create somebody with an extra digit on their fingers, which of course we know that happened in the past, and at which point does that make that person cease to be human? Of course, I'm not sure whether we know the answer to that question, but this discussion is about the state of the art methodologies, and there are many, to try and change the genetics of human beings. Am I right?
Absolutely. And it works hand in hand with the toolkit from Dr. James Giordano, who basically said that they were going to be doing this, and it was ready for prime time. It's now. It's not anything in the future. It's going to be happening before our eyes. NASA, in their Strategic Warfare 2025, said that they would be capturing and torturing humans on TV primetime. You can, in a way, that was old. That document is very old. I don't know how many. It's at least a decade, if not 2 decades old. This is capturing and torturing human beings. This is a form of torture and capture, and it is going to go prime time. It's already started. People that have been hit. I know people that have been hit. So this is in play. We know that. So it's up to us. I mean, we're behind the curve because they know what they're doing, and they have sophisticated equipment, and they know the plan. We knew generally what the plan was, but we didn't know the specifics. I think this gives us a lot of information to chew on and to learn about. Does everybody have to do the deep dive like I'm doing? No, but at least have some understanding about it so that you can take and make wise decisions and take precautions. It's precarious times.
Okay, that mostly sums up the questions, except that there are still some questions outstanding about this issue of venom from vipers and snakes. People are kind of, I think we're all a bit shocked at the inclusion of that in all kinds of remedies now, and somebody's even asking, is it in homeopathic medicine? Question about any other comments you want to give us on the issue of snake venom before we go out here,
The only thing that I would really recommend is if you're putting anything into your body, check the ingredients. Now, homeopathy is a little bit different, although I don't know when it comes to toxins. I use homeopathy, but I don't know about toxins. But any other thing, I would just say, get the professional insert and read. And if you don't know a word, I tripped on this just because I didn't know the definition of 2 words. And then I fell down this rabbit hole for a whole day and just went. It was like the Lord just was peeling away the layers of this program to give us some more insights and details into this. I don't have any more information except that I can tell you that since World War II, everything from rockfish to every type of the most lethal toxins around, they've been trying to get into us. And now this turns up, and there's links between the SARS-CoV-2 and rattlesnake enzymes. So we just have to be heads up, and I could use your help. I mean, if you guys see an article that pertains to what I'm talking about, please send it. Please don't overwhelm me. My poor inbox might not be able to take it, but if you see something, it's going to take all of our eyes, and it's going to take all of us to share notes to get through this.
Yeah, just in that vein, Celeste, there's a number of questions about, is it in supplements, is it in this, is it in that, or in organic products, etc., or in items with nanoparticles, etc. But at the end of the day, fair to say, I suppose really that this is a we are living in what amounts to a monstrous experiment, and those of us who are the subjects of the experiment, i.e., the humans, are looking all the time trying to discover what is being perpetrated on us next. And you know, you're one of the people who is helping us in that journey, but we don't know all the answers, do we?
No, we don't know all the answers. And it's incumbent on you. I don't know what you eat. And would never have the time to look up the whole global population, what they're eating, and be able to tell you on the label. And then if you have a label one week, there's so many mergers, the next week it could change and they wouldn't tell you. That happened to me back. I learned my lesson back in the 80s with IMs, and my dog got sick. And I contacted IMs and found out they had been bought out by another company, changed the whole recipe, if you will. And I didn't know it. And so my dog got sick. So you can't really even trust a label. If you don't understand the word, you're going to have to look it up. Yes, it's time consuming. Is it a pain in the neck? Yeah, but it just depends on to what level you want to have some security on what's around you. Definitely right now, the pharmaceutical industry is suspect. So I would not, like when they say, well, you can't know for 75 years. Well, most of us aren't going to be around for 75 more years. So then you have to say, okay, I can either go along with the program or I can oppose the program, of which then there's going to be consequences. And can I do that? Do I have the internal and spiritual fortitude to survive that?
Okay, that sums up the questions I've got, Celeste. If you're concluded, maybe we should, before we switch out, unshare your screen before we...
I have 4 more slides. We got
Oh,
to
that's
get
good.
Adam
Okay.
17.
Oh, okay. All right. There's more.
There's more. As Dr. James Corridano just said, and there's more. Yay. Yay for us. Okay, we're almost done. There's only 4 slides. So it's a disintegrin and a metalloprotease, also called TACE, which is the tumor necrosis factor converting enzyme. And I have a picture of Adam 17 right here. It utilizes a conditional knockout. That's the genetically modified mouse line called Adam. I won't go into there. And there's actually an international knockout mouse consortium. This is the gene edited humanized mice. And with a high throughput mutagenesis project to interested scientists. Male and females underwent a standardized phenotypic screening to determine the effects of the deletion. Oops, I'm doing this wrong. Whoops. TACE is a palliative treatment, which means it's not going to cure your cancer. TACE is used to control tumor growth and shrink the tumor. If you're waiting for a liver transplant, TACE can be used as a bridge. It is minimally invasive procedure performed in an interventional radiology to restrict a tumor's blood supply. Small embolytic particles coated with chemotherapeutic drugs are injected selectively through a catheter into an artery, directly supplying the tumor. Now this is, if you think that chemotherapy is the right way, this would be considered a medical therapy or option if you had cancer. This kind of shows you the TACE cycle. And then that's it. And coming up next, next time when we dive in, there is this occultic part. And so I am going to dive into that. I just thought that right now we just had a big enough or a more challenging trying to just understand these new words, these new technologies, what we're facing. This is much more complicated than the hydrogel and the quantum dot. So at least we sat down, we had the discussion. I will be getting this up. It'll be at Celestial Report, possibly YouTube. And also I will be making the document available at shepherdsheart.life. It might take me a day or two, depending on my schedule. But I just thank you for your interest. Thank you for your patience. And thank you for me stuttering over big, long words that I don't understand. And believe me, I really, even up until this morning, I was busy trying to simplify these words so to bring it down to something that you could understand. But in some of them, I didn't even understand the process. Most of it I did, but some of it I didn't. And at least you're aware that it's there and you have a reference that you can go back to. So I just pray that wisdom as you go through this. Don't have a spirit of fear, but of power, love, and a sound mind. And that will get you through. And just be very careful with purity and trying to keep yourself human and do actions of in the spiritual realm, praying, scripture reading, things that build you up and fortify you for these days and times. And, just be a human, give a smile, use your talent, use your passion to help other people to be strong during this time. And then also be that shoulder when somebody needs it, because that also is being human. So thank you very much for your time, and we'll see you next time with the occultic dimension of this whole Operation Adam.